Hello, everyone. Welcome to Examining Evolution. I am here with Dessel Drayce, and we're going to have a talk about abiogenesis. And I'm really excited about this because Drayce knows a lot about this topic. So,
Thanks for
Drayce,
talking me up.
yeah. And so, anyway, I'm looking forward to this. And so, how do you want to start? Do you want to start by telling everyone like a little bit about yourself and your interest in abiogenesis?
Okay, before we went live, you basically asked me like what got me into this whole thing, and so as I was saying to you before we hit record, what it was is basically I belonged to a fundamentalist church when I was very young. I ended up leaving that church, but I was really confused about what I believed in, and I wanted to figure out like how the world really was, and so like I got really into philosophy of religion and into apologetics. And so with that though, because God is used as an explanation for so many different things, that in turn led me to have to it forced me to get into so many different subjects. And eventually the emergence of life was one of them. And when I started to read about the subject, there are just like so many interesting things. It's like a highly interdisciplinary field where you have to learn about geology and chemistry and biology, and just so many different topics that it's just really fascinating. And because it was so fascinating, I ended up getting hooked. If you have any initial questions, before I dive into the material that I have, I'm happy to answer them. But basically, what I've done for this evening, I don't have a laptop anymore. So I'm basically operating on a cell phone where I can't share my screen. So I don't have any pretty pictures to show anybody tonight. But I do have material that I put together. And the point of this material is to argue that contrary to what you usually hear from creationists critiquing abiogenesis, that there has been substantial progress made in terms of understanding how life emerged on Earth. That is not to say that every single question about this or every single problem has been answered. No, that is not true. There's still a lot of work to do. But in terms of significant advancements that have been made, I think that there have been a bunch. And I kind of wanted to get into explaining why I feel that is.
Very good. So everyone, Drayce is going to give a presentation, and then we're just going to talk about and just discuss his presentation. So go for it. I'm looking forward to this.
Appreciate you. And so basically I want to begin with what I consider to be a mainline creationist critique of abiogenesis. And so I have this like quote that I wrote here. Like I'm not quoting anybody in particular. It's just the things that I would normally hear from a lot of people who are very critical about the field. And so basically it goes like this. It says, origin of life research has made no progress in over 70 years. There are many problems showing how natural, random, mindless processes cannot lead to life. There are problems of carbon and nitrogen fixation, problems with the synthesis of the required building blocks, the problem of homochirality, the problem of information, chicken and egg paradoxes, the water paradox, problems with waste removal, problems with concentration and polymerization, error catastrophe, the tomb problem for membrane transport, and the problem that the natural world only decays, degrades, or reduces in complexity. It never builds up complexity on its own. And with that, you'll hear people refer to things like Spiegelman's monster and stuff like that. We'll talk about that a little bit. This is especially true at the origins of life, where scientists cannot appeal to natural selection in order to optimize living processes. Moreover, the experiments conducted in origins of life research do not even count as legitimate evidence, since they carry out their work using purified industrial materials that are not found in nature. Even the term preabiotically plausible is hopelessly subjective, since the initial conditions of the early Earth are unknown and enjoy no widespread agreement among geologists. Scientists are baffled by all of these problems, and as time goes on, we find that the situation and the big picture only becomes more and more complex. And so that's what I usually hear from a lot of creationists about this field. And so like My initial feelings about this, just to give like a summary of what I make of this critique, is that a lot of the times I think that there's a lot omitted about the actual research. And we talked about talking a little bit about The Stairway to Life by Tan and Stadler. And there are several cases in that book where I feel that, because the book was published in like 2020, and they'll talk about certain issues in abiogenesis research, and there'll be all sorts of papers and experiments that are relevant to the question that they're considering, but they don't mention them at all. And I think that if you're going to criticize, a field or a theory, that you have this responsibility to critique it in its strongest light. You shouldn't omit evidence that's been put forward. You shouldn't criticize straw men. And another thing too is that they will, in my opinion, like they will choose particular things to criticize. That the way I kind of look at it is if like a kid walks out on the playground and they want to establish themselves as like the biggest, toughest guy on the block. And so they see somebody who's like really athletic and strong, and then they see this like little wimp that's like crippled in a wheelchair. And so in order to prove their point, they go up and they punch the kid in the wheelchair in the face, and then they declare themselves to be like the bully of the playground. And I just don't think that that's a legitimate way of going about things. Like you should be, if you're the true player in town, it's the strong person that you should be concentrating on. And I think that that's avoided a lot of the time. And so I wanted to really get into this critique, and where I wanted to start here is this idea of abiogenesis being this purely random event, because that's not true. It's especially not true for the theory that I'm most partial to. I support the idea of the emergence of life occurring at alkaline hydrothermal events. And so one of the bedrocks of this theory has to do with energy gradients and non-equilibrium thermodynamics and the idea of dissipative structures. And I'm going to explain what all of that means. But basically, I want to get into, like, for example, how certain other natural systems emerge by these kinds of processes. And this is a deterministic process. It's not random. What's going on here is that these structures that you see in nature are driven or forced into existence, by these kinds of like energy gradients. And so to start here, I wanted to focus, and I'm going to draw this into the question of the emergence of life. So you might think at first, like, why is he talking about this? Because it's relevant. It's based on the same principles. And so I wanted to start with, like, how is it that certain other non-equilibrium dissipative structures form? And the example that I wanted to focus on is a hurricane. So do you know, just to ask you, like, do you know very much about like hurricane formation, about like how these storms form, and like how they end up displaying this organized pattern and structure? Okay. All right. So I have this quote here. It's still one of the best examples that I've seen. It's written by Niall Shanks, who wrote this book, A Critique of Intelligent Design, called God, the Devil, and Darwin. And in chapter 3, Thermodynamics and the Origins of Order, he talks about the formation of hurricanes. And so this is what he says, and I'll draw it into the emergence of life. So he says, quote, "To get a hurricane, you need several things to come together, as natural mechanisms operate in accord with unintelligent natural laws during hurricane season. The ocean must be at least 27 degrees Celsius. You need a latitude of at least 5 degrees north or south of the equator. You also need a region of low pressure at sea level. Hurricanes, though undesigned, behave like heat engines. The ocean, warmed by the sun, is the heat source. There is a temperature difference between the surface of the ocean and the upper atmosphere, where it's much colder, and hurricane intensity reflects this temperature difference, as well as other factors such as pressure differences. Here's how the hurricane forms without the help of intelligent design. A region of low pressure draws in moist air from surroundings at higher pressure. This causes moisture in the air to condense, and the water, in changing state from gas to liquid, releases thermal energy as latent heat. The resulting warm air rises, drawing in more air from below, and hence from outside the immediate region of the forming hurricane. The water in this air condenses as well, releasing more heat. In this way, the eye of the storm forms, and the hurricane becomes a self-sustaining structure whose existence depends on energy flowing through the system and getting redistributed in the process. The spiral patterns shown by hurricanes result from the operation of Coriolis forces, which do not exist at the equator, hence the need for a latitude of at least plus or minus 5 degrees. The entropy requirements of the second law, that's the second law of thermodynamics, are such that the orderly motions and structures in the hurricane must be more than offset by disorderly, incoherent motions elsewhere, and anyone living in a coastal community where a hurricane has made landfall knows exactly what is meant by way of the incoherent motions of matter involved in settling the entropy accounts to satisfy the requirements of the second law. Landfall also disrupts the hurricane mechanism because of friction and a lack of moist air. In a sense, it is starved out of existence by getting cut off from the energy source that powers it. In the hurricane, there is a localized decrease in entropy as structure and pattern emerge. But this decrease is more than offset by increases in the entropy of the environment with which the hurricane interacts. The second law is satisfied. Creationists are simply mistaken that all natural, unguided processes must go downhill, unquote. And so people like Schrodinger long ago pointed out that life acts in much the same way. In order for us to perpetuate ourselves, we need to take in energy, which we use to perpetuate ourselves forward. And the second we stop doing that, we start to break down. And so they refer to this as like dynamic steady state. And that's what living organisms are. And so the idea here is that there are these mechanisms where there are these energy gradients that dissipate energy, and that energy is used for the production of structure and order, which emerges through that process. And it's a deterministic process. It's not some random thing. It's not just random molecules bumping around. These structures are forced into existence. And a hurricane isn't just the only example. They give many others, like convection cells, tornadoes. There are these things called, they're less interesting maybe, but this one called ice brinicles that have this very regular pattern about their formation. And so there's a lot that's discussed in this field of non-equilibrium thermodynamics. And this is the basis for this theory of how life came into being. And so I just wanted to put that out there that when abiogenesis researchers who support this particular theory, the alkaline hydrothermal vent theory, talk about the emergence of life, it's not a random matter. You know, the way that they might describe it sometimes is that, you know, considering these how the second law works, it's kind of like the universe really wants to relax, and it has all this pent-up energy that it's trying to do away with. But in order to relax, almost kind of ironically or paradoxically, the best way to relax is sometimes to build up complex things that dissipate out even more energy than was previously being dissipated before. And that's how these structures emerge. And so I'm going to give one last quote here about this topic, and then maybe pause for a second if you have any questions about it. So this is from a paper published in 1994 by Schneider and Kay, entitled "Life as a Manifestation of the Second Law of Thermodynamics." Quote, Schrodinger's equally important and less understood observation was his order from disorder premise. This was an effort to link biology with the fundamental theorems of thermodynamics. He noted that at first glance, living systems seem to defy the second law, as it insists that within closed systems, entropy should be maximized and disorder should reign. Living systems, however, are the antithesis of such disorder. Schrodinger solved this dilemma by turning to non-equilibrium thermodynamics. That is, he recognized that living systems exist in a world of energy and material fluxes. An organism stays alive in its highly organized state by taking energy from outside itself, from a larger encompassing system, and processing it to produce within itself a lower entropy, more organized state. Schrodinger recognized that life is a far from equilibrium system that maintains its local level of organization at the expense of the larger global entropy budget. And he finishes here, in the section of the paper entitled The Origin of Life, they write, quote, "The origin of prebiotic life is the development of another route for the dissipation of induced energy gradients. Life, with its requisite ability to reproduce, ensures that these dissipative pathways continue, and it has evolved strategies to maintain these dissipative structures in the face of a fluctuating physical environment. We suggest that living systems are dynamic dissipative systems with encoded memories, the gene with its DNA, that allow the dissipative processes to continue without having to restart the dissipative process via stochastic events. Living systems are like sophisticated mini tornadoes in this respect, with a memory, its DNA, whose Aristotelian final cause may be the second law of thermodynamics," unquote. And the last thing I'll say here before, basically asking you if you need some clarity on what I'm talking about, is they do emphasize that this is a necessary but not a sufficient condition for life. So this is a fundamental aspect of what we're talking about here, but there's still much more involved. And so I've said all that. It's a bit of a mouthful, but do you kind of understand what's going on here in terms of like the buildup of these energy gradients that need to dissipate themselves, and this occurrence of this free energy is being harnessed for the production of pattern, structure, and order?
Yes, although I think there's a big difference between a hurricane and a cell and the way that it's harnessing energy, right? So, I mean, I want to hear your whole presentation before we talk about it, because I know, one of the things in Stairway to Life is talking about this issue of harnessing energy and, the issues with ATP synthase and needing to exist, but it can't exist without ATP.
Right.
And ATP has to create it. So, and this is complex machinery. It's not just a wind and pressure system.
All right, it's highly complex, agreed. Okay, all right, yeah, we can definitely get into that, particularly that issue of energy harnessing. You're right, that's a very important issue in abiogenesis. Okay, so I'll continue on with my presentation, and then we can talk a bit more after.
Sounds good.
So what I want to talk about next here is, one of the criticisms that's talked about a lot in that book, The Stairway to Life, is they make the claim that that the term prebiotically plausible is subjective because there's no wide agreement on what the Hadean environment was actually like. And that's very true, that there's a lot of contention, like, was there land or no land? People generally agree that there was CO2, but they disagree about how much CO2. What was the salt content of the oceans like? What was the exact atmosphere, what were the atmospheric constituents? And so, there's a lot of disagreement about that, but there are several lines of agreement about certain fundamental things. And with respect to those things, those are all that I would need to proceed with an alkaline vent theory. And so I wanted to focus on those things that we have good grounds to believe in. And I don't want to make my argument on the grounds that, well, nobody has a problem with these things. They all agree that this was going on during the Hadean, even though that's true. Sorry, I got this notification popping up on my screen.
During the Hadean, what does that mean?
So the Hadean aeon, so we're talking 4.6 to 4 billion years ago, which is when.
Okay. Gotcha.
Yeah.
Mm-hmm.
So that's like the first aeon of Earth. It involves like Earth's planetary formation, and then going on to about 4.0 billion years ago, where it connects up with the Archean aeon. And so there's a lot of disagreement about what the Hadean looked like, but there are certain points of agreement. And like I said, I don't want to rest my case to you on the mere fact that everybody agrees about these things. I wanted to present you the evidence for why I think there's good reason to believe that the Hadean looked at least this particular way. And that's not to solve all of these contentious issues. It's just to focus on what we can know beyond a reasonable doubt about the Hadean. And so what I want to focus on here is the existence of an ocean, the existence of CO2, And I'm going to focus on, like I said before, there is contention about like how much CO2 is in the atmosphere. That's like there is what I think some geologists would say is like a consensus nowadays that there was very high levels of CO2 during the Hadean. But there are several geologists that have really put that to the question, like geologists by the names of like Sleep, Zonley, Chrysanthemum Toten, Koranaga, and others. And so they argue that there are certain processes that would take CO2 out of the atmosphere. Like, for example, they point out that during meteorite bombardments, it induces this process called impact ejecta-weathering. And this weathering process shuttles CO2 out of the atmosphere. And so I wanted to kind of focus a little bit on that, as well as the existence of the oceans. We also need to talk a little bit about phosphate, nitrogen, and sulfur. And again, I think people might get a little frustrated with me here because they'll be thinking, okay, like we want to hear more juicy stuff about like the synthesis of these organic molecules and their polymerization and like life, like all the particular aspects of life. We're definitely going to get there, but really quick first, we need to talk about the bedrock, and I mean that in quite a literal way. Like we need to talk about the bedrock of what was there in the Hadean to make sure that everything that we proceed to talk about isn't just a figment of our imagination. And so let's see here. All right, so I'm going to quote from a paper published in 2021 by Miyazaki and Koranaga, and they write, quote, ancient geological records suggest that by approximately 4.3 billion years ago, the Earth's surface had become similar to the present-day environment with oceans and granitic crust. Hadean detrital zircons, enriched in heavy oxygen and yielding low melting temperatures, indicate that liquid water was already present at the Earth's surface. They write this in an article entitled "A Wet Heterogeneous Mantle Creates a Habitable World in the Hadean." And so the important thing here is that they're arguing for the existence of an ocean based on the surviving zircons that we have from the Hadean, which is pretty much the only thing that survives from the Hadean. And so by analyzing these zircons that we have, they've come to the conclusion that there was liquid water present in the Hadean. And that's really important for the model that I'm supporting, that there were these alkaline hydrothermal vents. Once you have an ocean, once you have bodies of water, you're basically guaranteed hydrothermal vents based on how they form. They form through this process called serpentinization, which I'll get to momentarily. Now, they base this on the analysis of the zircons, and one of the main guys who's analyzed the zircons is this guy, T. Mark Harrison. And in this paper, I think it was written in 2009, he writes in this article entitled "The Hadean Crust, Evidence from Greater than 4 Billion-Year-Old Zircons." Quote, "We know that these ancient zircons yield relatively low crystallization temperatures, and some are enriched in heavy oxygen, contain inclusions similar to modern crustal processes, and show evidence of silicate differentiation at approximately 4.5 billion years ago. These observations are interpreted to reflect an early terrestrial hydrosphere, early felsic crust in which granitoids were produced and later weathered under high water activity conditions." They have this section of the paper entitled "The Hadean Water World Hypothesis," in which they give a bunch of evidence for the existence of an ocean. I'll just quote a couple of them because I don't want to spend too much time on this. I want to get to the juicier stuff that I think you and your audience will be much more interested in. And so, for example, the inclusion mineralogy within zircons is consistent with their formation in hydrous, meta, and paraluminous magmas, in the latter case implying both the surface origin of the protolith and the presence of liquid water and sedimentary cycling. Zircon crystallization temperatures cluster near minimum melting conditions, indicating a narrow range of magmatic environments close to water saturation. Plutonium-uranium variations in zircons are consistent with fractionation occurring in aqueous fluids. And so they're building a case here based on the evidence of the zircons that we have liquid water. And so a lot of people are saying that by 4.2 billion years ago, we've got an ocean in the Hadean. And so that's really important for the model that I'm going for. With the existence of the rest of this stuff, and I'm going to try to make this really quick here, CO2, the reason why people believe CO2, like there's a lot of CO2 in the atmosphere, is because as the Earth cools and these magma oceans cool, they degas. And the less dense material, such as CO2, gets shunted out to the upper atmosphere. But a lot of it also gets trapped inside the mantle. And so Koranaga and other geologists have talked about, these kinds of mechanisms that can shuttle CO2 out of the atmosphere. Even on their model, though, by the time you get to 4.2, 4.1 billion years, they start to converge on a high CO2 level. And more importantly, there are a series of papers by this researcher by the name of Lupton. And the first one that he published was in 2006, studying extant hydrothermal vents in the ocean. And what he found at a number of them was that around the site, there's actually liquid CO2, highly purified liquid CO2, purified up to like 98%, that's bubbling up from the mantle, from the sea bottom. And so you get CO2 that way as well. And so even in a worst case scenario, I think we can confidently say that even if Koranaga, for example, is totally right, that CO2 is being shuttled out of the atmosphere at insane rates because of meteorite and bombardments and everything like that. Nevertheless, there's a lot of CO2 that's still bubbling up from the ocean bottom around these hydrothermal vent structures. And the reason why I'm focusing on things like CO2 is because life is composed of six really important elements. They abbreviate it by the acronym SHNOPS. So you're talking carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And you need all of those in order to produce everything that you need for life. And so I focused so far on carbon. You have oxygen already. Nitrogen is something that people like Michael Russell have focused on through lightning, for example. The nitrogen gets absorbed into the oceans in the form of nitrate and nitrite, and so you get your nitrogen source that way. Phosphorus is like a big thing in abiogenesis research because people are really skeptical that you have a lot of available phosphate during this early period during the Earth's Hadean aeon. But there are important articles that have been written, some of them really recently, by this researcher Craig Walton, and he points out that you'll find phosphate, for example, in olivine, which is directly relevant to hydrothermal vents. That's basically the rock that's undergoing serpentinization, which is what's creating these hydrothermal vents. And so the last here, the hydrogen and the sulfur, that serpentinization creates that. And I've mentioned this process several times, so what the heck am I talking about? Basically, there are these really dense materials in the mantle, and they don't like to be near the surface, but plate tectonics can force them up toward the surface, and as they go up there, they go through all these crazy metamorphic changes, and once they reach the ocean, they undergo this process of serpentinization that produces a bunch of minerals that end up constituting these vents. That's how you get your hydrothermal vent, and it also ends up creating hydrogen sulfide. And so right there, you have, in this one environment, you have all 6 elements that you need. And based on the evidence that I've referenced over the course of this, I think that we can say beyond reasonable doubt that this environment did exist in the Hadean. This is again something that most everybody agrees on, but I didn't want to base my case to you just based on that. So we can turn from that and try to start getting into more interesting things. I still need to talk to you a bit about these vents. Like, why is it that they can produce life? What is it about them? How is it that they work, that they can work in such a way that life emerges from them? And so basically the vents have a mineral constitution, and something that abiogenesis researchers will say is you still go to the local pharmacy to get like your magnesium and your zinc and your iron and all this stuff that's necessary for your body to function properly, and like something like 40 to 50% of your enzymes in your body are metalloenzymes. they work by having these like metal centers. And this is really important for things like respiration and redox chemistry. And all of these minerals and metals are there at the vent. And so the initial supposition was, well, if your body uses these, these inorganic metals in order to accomplish these tasks, is it the case that the metals by themselves could catalyze the same reactions? And so this was a kind of like metabolism first approach to abiogenesis. And way back in the day now, about 2008, Leslie Orgel, a famous abiogenesis researcher, wrote this paper that basically entitled The Implausibility of Metabolic Cycles for Emergence of Life. It's not the direct title, it's just close to it. He's really skeptical about this being the case. But since then, there have been so many different experiments done using things like iron, calcium, copper, nickel, molybdenum, tungsten, palladium. And these things are able to catalyze a huge number of the initial metabolic steps that you need. Like Muchowska and Moran show 6 out of the 10 steps of the acetyl-CoA pathway, a bunch of the steps of the reverse Krebs cycle and the citric acid cycle, all catalyzed simply by iron and similar metals directly themselves. And when you take a look at the way that that the inorganic centers of these enzymes are structured, they look exactly the same as the structure of these metals and these minerals that you find there at the vents. And so they've been able to show now that by using them, you can catalyze all of these metabolic reactions. In the case of palladium recently, Harpreet Kaur showed that you get the whole cycles. She's got a paper that's coming out pretty soon, and she recently gave a presentation on this. And you do it under like you can do it at room temperature. You don't need like extreme conditions for any of it, and you drive a robust amount of our metabolic cycles using these transition metals. So let's see here. So I've talked about some.
Drayce, let me just tell you, you told me to interrupt you when you go for about 30 minutes.
And I am at 30 minutes.
And so that we could have a discussion, but I feel like you basically just started your presentation. Yeah. So we haven't even begun to like get into what you want to talk about. So I'm just going to let you know, I can let this stream go for as long. I want you to have the opportunity to share all the information that you want to share. So if you're not in a rush, I'm not in a rush.
I appreciate that very much. Okay, thank you.
And I just have to say, I want some feedback from the audience. Isn't Drayce amazing? He knows so much about this topic. It's great. I really appreciate the work that you've put into this.
I appreciate that, and I'm also like I came here for 2 main reasons, which was to try my best to like try to be able to convey this in an understandable way. That's usually something that I try to challenge myself to do, but also to get criticism, because I mean, like, what better place to go for criticism of my theory than people who absolutely can't stand the idea of it. So like I think I'm hoping that over the course of this presentation, some people can at least give me some valuable criticism. I'm not going to sit here and say that I think that everything everybody's going to say to me I'm going to think is a legitimate point, but maybe some interesting things get brought up over the course of it. So we'll see. So some last things about like these vents and how they work. So one of the ways that there's an analogy here between the way that these vents are structured and the way that you yourself actually harness energy. So there's a couple of things here. Like during the Hadean Aeon, there's this particular kind of clay mineral surrounding hydrothermal vents called Fougerite. It's an anionic clay, and it is a layered double hydroxide. So it has these layers, and there's small bits of water in between, and it acts peristaltically, which means it acts as a pump. So you're talking about that ATP synthase before, that really complex pump that's pumping protons through for the production of ATP. this clay is really freaking weird. They're using it now in like technological applications and also for like pollution reduction and all this other stuff, because it has these really weird technical aspects to it, where like it imbibes a bunch of things. It can take in like phosphate and all sorts of other stuff. Like I said, there's small bits of water in between there that's relevant to like the water paradox. We're talking like much reduced water activities in between these layers. But it also acts peristaltically, like pumping these protons through, and it's also conductive. It conducts these it conducts things with charge through to the other side, so like that's one thing about like the way that this works. But the way that your body works in terms of producing energy is you have what's called the electron transport chain. And it works with this pH gradient, where the electrons build up these protons on one side and shuttles it across this chain to the ATPase, to the ATP synthase, pump, and it passes through for the production of ATP. And what you get there at the vents, right there at the interface with the wall, is precisely the same thing, where you have the you have CO2, like we talked about on the one side, which acidifies the water, so it's a reduced pH. But on the other side, you have this alkaline fluid of hydrogen sulfide that's going up through the vent, which is a much higher pH, and so you have this pH gradient between the two. And that is the mechanism by which, even in your own body, the protons are passed through for the production of ATP. And so the idea is that, the events itself give you a natural pH gradient for this to occur, for the protons to pass through for the onset of the production of organic molecules. And so they've actually been able to do this now. In 2020, there was this group led by Hudson, where they were able to use a pH gradient to reduce CO2 using hydrogen to produce formate, an organic molecule. And so that was like a proof of concept that was actually really contentious in the literature for a long time. There's this critic of alkaline hydrothermal vents named J. Boz Jackson, and for a while there he was just like, no way, this will never work. But over the course of time, experimentally, they've shown that yeah, it works. So you have this pH gradient that works in this way. It's very analogous to the way that your body manufactures ATP, and it also has these other gradients. Like there's this thermal gradient that hydrogen sulfide fluid is very hot. Well, it's much less hot than certain other events. It's you could say the average temperature they give for is like 120 degrees, and so there's this heat source there. But along the edges of the vents, it's much colder because it's interacting with the colder ocean water. So you have this like hot source going up through it, and then around the edges, it's much colder, like 30 degrees or so Celsius. It goes down to like room temperature. And the reason why that's important is because like you're synthesizing these organic molecules by this catalytic kick of these higher temperatures that are speeding up these reactions. But if they stay there in that hot temperature, that's when you start to hear a lot of these complaints about them degrading. They degrade at a higher rate in higher temperatures normally. But if it's the case that the kind of flow that goes on in an alkaline vent pushes those organic molecules out toward the colder regions, which we'll get into, then they stabilize in those colder regions. They're not in that hot area anymore where they're going to be degrading rapidly. So I just want to check here to make sure that I've covered like at least like the basics of what we need to talk about in terms of like, the vents and how they work. Yes, it looks like, last thing I'll mention here is when they synthesized Fougerite, the clay, what they were able to do is they attached a light emitting diode to it directly, and it was able to power the light bulb, and it kind of reinforces the point of how it's electrical. And this is one of the main points that you hear from the guys who support this theory. is that life is electrical. It's that like something that your friend Saigar says in an interview. He says, chemistry doesn't act this way. And so that's one of the reasons that led him away from thinking that abiogenesis is like a legit field. Is he knows chemistry and he knows that chemistry doesn't act this way. But the supporters of this theory, which is a primary theory in origin of life research today, have been saying the same thing for a very long time. That life is much more than just mass action chemistry. It transcends chemistry because we're talking about non-equilibrium thermodynamics. We're talking about life as also electric, something that Galvani found out a really long time ago when he started poking that frog with an electric prod. And so that's enough about these vents. And you have the makeup now. You understand what's going on. You understand that, like, we're talking about metals catalyzing these reactions. There's this pH gradient there that works very similar to the way that your mitochondria produces energy with the electron transport chain. The way that Fougerite acts as like a pump and stuff like that. So, like, what do we get? Like, let's get to the interesting stuff now. What are we synthesizing, and how do they complexify? Do they complexify? Is there experimental evidence that they do so? And so what's been shown experimentally so far is that you get pretty much everything that you need already, like in terms of being experimentally shown. There's a little bit to do with nucleotide formation that I'm a bit unhappy with, but it's because I want to see them in higher yields. But with that as an exception, we're talking about amino acids, carbohydrates, lipids, and informational polymers, and we've already been shown experimentally that we get all of that stuff. And so I want to go into some of the research that's shown about this, and to start with this metabolite, pyruvate, which is involved in it's along the pathway to glucose metabolism in your body, and once you get pyruvate, you start to get things like amino acids. And so there's this article that was published in 2020 by Prainer. It's called A Hydrogen-Dependent Geochemical Analog of Primordial Carbon and Energy Metabolism. And this has to do also with, it's important to talk about things like fixing carbon, fixing nitrogen for the production of these organic molecules, using these elements to produce the organic molecules that we need. And this article addresses both the synthesis of pyruvate, this metabolite, and showing experimentally that you can fix carbon for the production of these organic molecules. And so just to read the abstract to you quickly, quote, hydrogen gas, H2, is generated by alkaline hydrothermal vents through an ancient geochemical process called serpentinization, in which water reacts with iron-containing minerals deep within the Earth's crust. H2 is the electron donor for the most ancient and the only energy-releasing route of biological CO2 fixation, this acetyl-CoA pathway. At the origin of metabolism, CO2 fixation by hydrothermal H2 with serpentinizing systems could have proceeded in patterned biotic pathways. Here we show that 3 hydrothermal minerals, greigite, Fe3S4, magnetite, Fe3O4, and awaruite, Ni3Fe, catalyze the fixation of CO2 with H2 at 100 degrees Celsius under alkaline aqueous conditions. The product spectrum includes formate up to 200 millimole, acetate up to 100 micromoles, pyruvate up to 10 micromoles, methanol up to 100 micromoles, and methane. And when we're talking about these synthesis experiments, what's relevant for the origin of life is you need micromolar to millimolar quantities. That's what you're really looking for in order to be able to actually go on and expect organic processes to proceed. If you have any less than that, that's no good. But if you have at least micromolar quantities, you're looking more for millimolar quantities in terms of nucleotides. And if you have molar quantities, that's more than enough. And so far we've shown some evidence for CO2 fixation, and I've shown you evidence that we can synthesize this metabolite, pyruvate. And from pyruvate, you start to get amino acids. So some evidence here for the synthesis of amino acids. There's this article published by Barge in 2019 using pyruvate. It's entitled Redox and pH Gradients Drive Amino Acid Synthesis in Iron Oxyhydroxide Mineral Systems. And so using pyruvate in iron oxyhydroxide mineral systems, they're able to synthesize alanine. And what they also found is that these gradients that exist there drive product selectivity. So do you know, for example, you might have seen him around, there's this scientist, a chemist, biochemist, I believe, James Carter. He's appeared on a number of shows, like on Standing for Truth.
Yeah, I know him.
Yep. One of the criticisms that he makes about catalysis is that you can have a catalytic reaction, but it can fail to be specified toward the exact products that you need. And so one of the important things about these gradients is that they can further specify the reaction to give you exactly what you're looking for, that there are environmental factors that will further specify a catalytic reaction to give you what you need. And so that was one of the things that they talk about in this paper. And another really important paper is my favorite abiogenesis paper so far. It's published by this guy, Hideki Fuka, and I'm probably even pronouncing his name wrong. I've never heard it pronounced out loud. But it was published in 2019. It's called Electrochemistry at Deep Sea Hydrothermal Vents, Utilization of the Thermodynamic Driving Force for the Emergence of Life. And he makes a similar point in there about how there are environmental factors, these gradients that you can use that will specify that reaction to give you what you're looking for. All right, so let's continue there. So we have pyruvate, this metabolite, we've got some alanine, an amino acid. You can look next to Hydrothermal Vents and Prebiotic Chemistry, a review, which is written by I think it's Maria Colin Garcia, published in 2016. This is a very helpful paper, and the reason why is because it's a review of a lot of the work that's been done over the past couple of decades about just what you're synthesizing under these environments. And so just to throw out there all of the amino acids that have been synthesized that are reviewed in this paper: glycine, alanine, aspartic acid, glutamic acid, serine, proline, leucine, isoleucine, valine, and threonine. And that was just, over the course of this paper and in 2016. There's been subsequent work done. That's a lot of amino acids that have been synthesized under these conditions. But I will admit to you that so far all we've got are some amino acids. We need them to polymerize into peptides, and this is only one category so far of the elements of life. Another really important.
Are those homochiral?
That's a point that we're going to get to shortly.
Okay.
So, with respect to the homochiral problem, I'll talk about that momentarily. But it's an aspect of dissipative systems like this, with the existence of this thermal gradient and the dissipating energy, the kind of flow and convective flow that it uses actually incurs what's called spontaneous mirror symmetry breaking. It's one of the kinds of theories that have been put forward to explain homochirality. And so what it does is it actually partitions enantiomers of a particular chirality into the relevant colder portions of the vents that we're talking about. And that's where further is going to take place in these much more stabilized, colder regions. And so now there's like this, I'll talk about this more momentarily. It's this process called thermophoresis. And all that we're talking about with this big word is the heat gradient. And with the existence of this hot fluid at the center percolating up, it creates this force and flow where these larger organic molecules they oscillate at first, and then eventually they start to polymerize, and then they shuttle off. They're actually, the larger they get, the more that they're selected under this kind of framework to shuttle off into the colder regions and get trapped there, because the flow is determined by their weight. And it concentrates them very heavily, and they polymerize and then get shuttled off to the outer edges and get stuck there. And it's fortunate that they do so, because it's much more stable there for them. They won't degrade in this hotter environment, toward the center with this hot alkaline fluid. They're now in the outer edges near to the ocean water, and it's much colder there where they can endure. And there's a bunch of other things as well that helps them to endure, certain interactions that occur between them, between the organic molecules themselves, that actually help to stabilize one another. And this was a kind of a drawback for abiogenesis research for a while, is that they were all considering their own respective, like, this world first and this world first. They're all considering like RNA in a vacuum or proteins in a vacuum, and not considering how when you synthesize these molecules, how these molecules are interacting with each other. And it turns out that they help each other substantially. They have symbiotic relationships with one another that help to stabilize both of them, and to, like for example, with peptides and RNA, to help onset further processes that are going to take place later on. This stability is ultimately what's going to lead to things like coordinated cellular purposes and to the onset of the genetic code. The last amino acid that I want to talk about is cysteine. This was in 2020, and I'm lying, there's one more, because one of the things, like with hydrothermal vents, like people will ask, well, why isn't life being synthesized today? And so like there are various reasons, like the fact that the ocean isn't as carbonic, it doesn't have as much CO2, so it's not drawing that in as much anymore. The fact that there are these ecosystems that live there at the vents that chew up anything that gets produced. But there's been a lot of articles that have been published actually verifying that there is limited abiotic synthesis of organic molecules ongoing at the vents today. And so an example of this was the amino acid tryptophan. In 2018, this researcher, Menez, identified tryptophan. There are distinct biosignatures that are left if something like an amino acid, such as tryptophan, distinct biosignatures that would be left if it's being produced by a living organism. It's one of the ways in which astrobiologists have really tried to distinguish, like if they're looking at a planet and they see certain molecules or features on that planet or on that moon, is it because there's life there, or is it just because there's a geochemical process producing these organic molecules? And so they've really had to work hard to try to distinguish between the two. And so there are certain biosignatures that will be left behind that they can pick up on. And if they're absent, they consider that to be evidence that it's being abiotically produced. And so in 2018, for example, tryptophan was identified as being abiotically produced. Abiotic synthesis of amino acids in the recesses of the oceanic lithosphere is the title of this paper. And also 2020, prebiotic synthesis of cysteine peptides that catalyze peptide ligation in neutral water by Foden et al. Another thing to point out here really quickly, the alkaline fluid that's percolating up through this vent, of course, highly alkaline. But the water, the ocean water on the other side, more acidic. In between these two, though, as they start to interact with one another, you get to areas of the vents in the colder areas that are more pH neutral. And that's where they're, again, this is the area that these organics are getting stuck through this process called thermophoresis. This process is limited to a certain number of vents. It doesn't occur at all of them, but it occurs at a bunch, particularly freshwater vents, any vent that doesn't have like a really high salt content. Another open question about the Hadean is like, how salty was the ocean? And so like, was it really salty? If it was, then thermophoresis isn't going to work in the ocean. You would need a freshwater system, something like a lake that's been created by like a meteor impact or something like that, where a hydrothermal system emerges underneath it. And in that kind of environment, this process of thermophoresis would be working. We'll get into that momentarily. So I've given you a bunch of amino acids. What about lipids? So like phospholipids, for example, one of the earlier examples that I was able to find on this was published by Hargreaves and Deemer, synthesis of phospholipids in membranes in prebiotic conditions going back to 1977. A recent one, synthesis of phospholipids under plausible prebiotic conditions and analogies with phospholipid biochemistry for origin of life studies by Fiore et al., published in 2022. This is basically working through things like Fischer-Tropsch synthesis which converts carbon monoxide and hydrogen into hydrocarbons in the presence of metal catalysts. Another paper here by Jordan, published in 2019, I think it was, Promotion of Protocell Self-Assembly from Mixed Amphibials at the Origin of Life. This is another thing where salt is an important issue. If you have high salt content, for a while people believed that you wouldn't really be able to form lipid chains. But what was later shown is that even in the presence of salt, as long as you have a heterogeneous chain, it'll work okay. It's just homogeneous chains of lipids that have more trouble forming and much higher salt contents. And so Jordan came around in 2019 and showed that you can form from these mixed amphibials, you can have protocell self-assembly. And an interesting follow-up paper to that was Sarkar et al. 2020, which focused on these mixed amphibials and found that if we're talking about like natural selection or their sustainability and robustness to environmental stress, these mixed amphibials are much more robust to environmental dangers. And so that is, I just want to make clear that I haven't totally bought into that. There are certain other approaches to creating a membrane that I think are really interesting. But these are synthesis experiments that have shown that you can get these kinds of lipids that will compartmentalize, and once you get them under proper concentration, like high enough concentrations, they automatically will compartmentalize. And Zostak and Budin in 2009 showed that not only can you get them up to high enough concentrations, but they'll actually encapsulate DNA once they do so. Let's move on. RNA and nucleotides. This is one area where I think a little bit more work needs to be done, because even though they have synthesized canonical nucleotides, like gone all the way there, like step by step by step, and gone all the way up to canonical nucleotides, that particular paper that showed that only gave us an 8% yield, which I think is kind of crummy. I want to see better than that, personally. But there's a lot of other stuff, like, for example, Akamura 2019, a one-pot water-compatible synthesis of pyrimidine nucleobases under plausible prebiotic conditions. So you get your nucleobases, and these one-pot experiments, you hear a lot from people who are critical about abiogenesis, about all this human interference that you need in order to make these syntheses take place. A one-pot experiment, though, involves no human interference at all. There's numerous different kinds of like no human interference type experiments. One pot is one of them. Another one that they call them continuous reaction networks. And if you go and you Google in one pot prebiotic synthesis or a prebiotic continuous reaction network, you'll see a bunch of articles pop up. A non-enzymatic analog of pyrimidine nucleobase biosynthesis by Yi et al. 2022. Cysteine and iron accelerate the formation of ribose 5-phosphate. Integrated synthesis of nucleotide and nucleosides influenced by amino acids. This was something I was talking about before, how these organic molecules interact with one another in very helpful ways. They help each other. Not only do they help for things like formation, but they keep each other stable. The cooperative interactions between them leads to further complexification down the road. Lipid-assisted polymerization of nucleotides by Olasagasti and Rajamani in 2019. A prebiotic synthesis of canonic pyrimidine and purine ribonucleotides by Kim and Kim. That's the paper I was complaining about. They get approximately 8% yield and they are operating at 125 degrees Celsius. That would be present at the vents with the alkaline fluid. Temporal nanofluidic confinements induce prebiotic condensation reactions in water. This paper is really important for a number of reasons, but this paper is a preprint. So this has not been peer-reviewed. The reason why I bring it up is because the authors, De Herrera, for example, has already given his presentation on this to the Euro Climate or Euro Planetary Society, or whatever the hell it's called. He gave a formal presentation where they're able to synthesize RNA under hydrothermal conditions, where the conditions are nano-confined water. The reason why this is important is because it had to do with the water paradox. And I'm going to rush on so that way we can get to these problems, because I know you want to talk about things like homochirality and the water paradox and stuff like that. And I'm just going to say 2 words about ribose and carbohydrates and stuff like that. The foremost reaction is one possible mechanism that's already been shown not only to be operative at the vents, but through a process of thermophoresis of this convective flow and this heat gradient, you can concentrate something like ribose over 1,000-fold. And that's so let's actually focus on that, right? Because Stairway to Life, one of the main problems it has is about concentration. How do you concentrate and polymerize these basic ingredients that I just gave you? And so I want to focus on this paper by Mast et al., published in 2013. It's entitled Escalation of Polymerization in a Thermal Gradient. And in the abstract, it says the following. For the emergence of early life, the formation of biopolymers such as RNA is essential. However, the addition of nucleotide monomers to existing oligonucleotides requires millimolar concentrations. Even in such optimistic settings, no polymerization of RNA longer than about 20 bases could be demonstrated. How then could self-replicating ribozymes appear, for which recent experiments suggest a minimal length of 200 nucleotides? Here we demonstrate a mechanism to bridge this gap, the escalated polymerization of nucleotides by a spatially confined thermal gradient. The gradient accumulates monomers by thermophoresis and convection, while retaining longer polymers exponentially better. Polymerization and accumulation become mutually self-enhancing and result in a hyper-exponential escalation of polymer length. A pore length of 5 centimeters and a temperature difference of 10 Kelvin suffice to polymerize 200 mers of RNA in micromolar concentrations. The probability to generate these long RNAs is raised by a factor of greater than 10 to the 600. Compared with polymerization in physical equilibrium. We experimentally validate the theory and the results confirm blah, blah, blah. And so I want to focus on that number for just a second. Stairway to Life, think about the numbers that it uses when it talks about probability. It gave you like 10 to the 70, 10 to the 90, that this is, you're much more likely to be able to locate like a single atom in the entire universe than for the probability of this happening. And the problems that I have with those kind of figures is that it's usually assuming some kind of random interaction, and that's why a paper like this is really important, because it shows that when you're not talking about random processes but deterministic processes, how much more the probability is raised. Just like with that hurricane, where you need very specific conditions in order for it to form, and it's driven into existence once you get those specific conditions. Once you have specific conditions that will drive these processes toward complexification, the probability toward things like polymerization becomes exponentially higher. So compare that 10 to the 70 or 10 to the 90 that you saw in that book, and now compare it to the greater than 10 to the 600 that you see right here. It's kind of like, like a midget trying to walk into a ring and pick a fight with Andre the Giant. So like at first, the 10 to the 70 or 10 to the 90 sounded really impressive, like, wow, it sounds impossible. But now you take a look at this paper that's giving you a figure of greater than 10 to the 600, that it will, because of the nature of this process, driving it toward these particular reactions, this kind of complexification. Now we're on the, another thing that it mentions in that paper, Spiegelman's monster, that's also talked about in that book. So they're talking about how, like, RNA, which is pretty much just considered, again, in a vacuum, will progressively simplify itself rather than complexify. But this particular kind of method of polymerization and concentration selects for the longer polymers, and it's those that end up getting shuttled toward the stabler regions of the vents. And so you're literally, the ones that are most likely to get caught and preserved are the longer ones, and it's promoting this polymerization to get longer and longer. And this was also shown in a bunch of other papers. Bosca 2007 started out with a model. Hadean 2009 that I referenced before showed this not just for nucleotides, but also for lipids. And then there is Hershey 2014 that did it for ribose, showing 1,000-fold concentration. So now we're on to homochirality, and I talked about this process of like spontaneous mirror symmetry breaking. And so for this, there's this particular researcher that I like. His name's Josep Ribó, and he's published a bunch of papers on this. And he's not the only one, but he's the one that I found most interesting. His papers are entitled Spontaneous Mirror Symmetry Breaking in the Limited Enantioselective Autocatalysis Model abyssal hydrothermal vents as a scenario for the emergence of chirality in prebiotic chemistry. That was published in 2013. Got a follow-up in 2019 in this book called Possible Routes to Homochirality in Prebiotic Earth or something like that. I can't remember the exact title off the top of my head, but the title of his paper is Chemical Basis of Biological Homochirality During the Abiotic Evolution Stages on Earth. It talks about the same process, about spontaneous mirror symmetry breaking leading to the onset of homochiral systems. There are other authors that have supported the same hypothesis. Karuna Karin in 2018, Spontaneous Symmetry Breaking in the Formation of Supramolecular Polymers, Implications for the Origin of Biological Homochirality. And then some of these models rest on there being like just some kind of initial imbalance, some ever so slight imbalance of a preference for one kind of an enantiomer. And so like there are a bunch of different ways if you are relying on a process that gets amplified from some small imbalance, there are a bunch of different ways in which that initial imbalance can take place. For example, like preferences in terms of like what a mineral catalyst will form. And so there's also with respect to like interactions between the molecules themselves, again, there's this paper by Armando Cardova, not your Sal, but Armando, and it's titled Plausible Origins of Homochirality in the Amino Acid Catalyzed Neogenesis of Carbohydrates. And so if you have these processes that give you an initial imbalance, that gets amplified through this system to give you a heavy chiral bias. And I'm not sure whether or not I believe that these abiotic processes led to a pure homochiral state. There's some phylogenetic evidence from George Fox, I believe, on the evolution of the ribosome that suggested that at its origins, we're not dealing with a pure homochiral system. And I'm not really sure which way to go about that. And there's a lot more that I need to read on this issue. But nevertheless, what these processes do show is that you can get strong chiral bias with respect to the way that these dissipating systems are operating. The water paradox. About this, there are two things: that water degrades things like RNA, and that it prevents condensation reactions for like polymers to form. And so there are a bunch of things about this system, and also again about the way that these organic molecules are interacting together that prevents degradation by hydrolysis and facilitates condensation reactions. And in order to understand how this works, we need to talk about something called water activity. So you have water, and say you take like one organic molecule and you throw it into a pool. That pool of water has absolutely nothing else to interact with other than that organic molecule. And so that water is going to break down that organic molecule as fast as water can break it down. But now let's say you take that water and you confine that water, you encompass it by some kind of nano-confining structure, a mineral system, like for example in the vents. Now that water is interacting with all sorts of other stuff, and there's much less water in that particular area. Because there's now less water, and that water is chemically bonding to all sorts of other stuff besides that one organic molecule, it's interacting with that molecule much less, and so therefore degrades it at a much more reduced rate. Likewise, when you have things like lipid compartments, that lipid compartment protects that organic molecule, a peptide or an informational polymer, from degradation by hydrolysis. And then there's another aspect, serpentinization, that we talked about before. This acts, one of the more interesting things that I've seen recently, the papers that are relevant about this, The Ambivalent Role of Water at the Origin of Life by Nascimento Vieira, published in 2018. The Effect of Water Activity on Rates of Serpentinization of Olivine by La Madrid in 2017. And again, that paper by De Herrera, that's a preprint. All of these are important, talking about what happens when you confine water under nano confinement such that it's interacting with other things and there's less of it, and how this bypasses the water paradox. And you can test that. Like, I could just tell you, like, okay, well, we're just going to confine it. and it's not going to be nearly as bad. And I could just assure you of that and give you no experimental evidence. Or we could actually test it to see whether or not it works. And so there's this, so I want to focus on this researcher, Alexandra Witcher, and she wrote her PhD thesis, Simulated Hydrothermal Vent Environments to Investigate Prebiotic Metabolism at the Origin of Life, in which she specifically tested whether or not you could get condensation reactions in water under these conditions. And she was able to, she was also investigating whether or not you could use acetyl phosphate as a primordial energy currency. And what she was able to do was demonstrate condensation reactions in water all the way up to adenosine monophosphate. And it was that adenosine monophosphate that later De Herrera used in his experiments in order to synthesize RNA under these conditions. And so that's how that the whole thing about the water paradox works, is that there are a number of interactions between the organic molecules, such as the lipid protection, such as the stabilization between peptides and RNA that helps to stabilize each other, and also the nano confinement of the environment that forces that water to interact with the surrounding environment and not chemically bond so much with that organic molecule. And likewise, there's less of it, for example, in the interstitial layers of that Fougerite clay, much less water. And I want to press that all of this is happening in the freaking ocean. Like, that was something that really confused me at first. Like, this can't possibly work. Like, you're literally telling me that, that you can reduce water activity, like all the way underneath the freaking sea. But when I looked into it, like, because we're talking about a vent structure, yeah, and we're talking about these layered clays, that only have very reduced amounts of water content in them. So I've talked about homochirality, the water paradox, and I'm going on like an hour here, so I'm going to focus on just a last couple of things here. Then I'm going to shut up and we can have a conversation. I apologize for going on so long. I thank you for humoring me.
No, you don't need to apologize. This is wonderful. Keep going.
Okay. All right, thanks. So I want to talk a bit about the origin of the genetic code. So focused on one of the really important papers on this that I found was the mutually stabilizing interactions between protopeptides and RNA by Moran Frankel-Pinter. And he talks about the symbiotic relationship that ensues between peptides and RNA, how they really help each other in terms of their stability. But another thing that was really important, you had Dimitar Kunev on this show a while back, and he has made what I consider now to be an enduring contribution to origin of life research, because he made this hypothesis about the existence of what he calls a bridge peptide. And it is what this is a peptide that is able to facilitate between the languages, quote unquote, of protein and RNA, and to allow for RNA replication. And so he hypothesized that you would have this bridge peptide that facilitated replication. And that was just a hypothesis when he came out with the paper. The name of the paper is a mouthful. It's possible emergence of sequence-specific RNA amino acylation via peptide bond intermediary to initiate Darwinian evolution and code through origin of life. Kunev and Gospodinov, 2018. And when he wrote it, it was just a hypothesis. And he was really interested in hopefully convincing somebody to do the experiment to see whether or not they could synthesize just such a peptide that would be able to facilitate replication for RNA in this kind of RNA peptide relationship. And that subsequently happened in 2022. Muller et al. In their paper, A Prebiotically Plausible Scenario of an RNA Peptide World, synthesized just such a bridge peptide. And so that's focusing there on this mutual interaction between them. And it also goes to this whole chicken and egg paradox, which came first. The answer there is that neither one came first. They both go all the way back to the beginning, because this particular environment synthesizes both of them, and they both have mutually stabilizing symbiotic interactions with one another, such that it facilitates further complexification, such as coordinated cellular purpose and the emergence of code. And let's see here. We're also, with respect to the code, I want to talk a little bit about the origins of the ribosome. There's this important paper by Jessica Bauman and Lauren Williams, The Root of the Tree, The Origins of the Ribosome, published in 2020. They structurally analyze ribosomes and come to the conclusion that that the ribosome evolved primarily through accretion. So you have this central peptidyl-transferase center that's responsible for peptide bond formation, and around that basic center accreted more and more and more material. That was their hypothesis. That was their theory based on their structural analysis of the ribosome. Now, again, you could test that. You can, for example, synthesize. You could basically throw away all the outer edges of it and just concentrate on that central peptidyl-transferase center to see whether or not, even getting rid of all of the other complex stuff, whether or not that center is still on its own capable of peptide bond formation. So there is this paper written, Origin of Life, Proto-Ribosome Forms Peptide Bonds and Links RNA and Protein-Dominated Worlds by Boas et al., published in 2022. They synthesized 13 different proto-ribosomes, basically working with the peptidyl-transferase center. They synthesized 13 different ones. 5 of them were fully functional, providing direct evidence for this idea that, yes, the ribosome evolved through an accretionary way, where the initial function is preserved right there at the center, even without all of the other complex array of things that eventually formed. And you see like the gradual complexification of it from like bacteria and archaeal ribosomes onto the eukaryotic ribosomes that have like all these like crazy spindles and stuff coming off of it. It has like, there are more extravagant structural features to the eukaryotic ribosome. And so I'm down to the last of it here, which is acetylphosphate as a primordial energy currency instead of ATP. And so in the paper, or in the book, Stairway to Life, the authors make fun of Nick Lane for suggesting this. They say, that's a really cute fairy tale, basically, is what they say. You can just imagine or fantasize that acetylphosphate can be used instead of ATP in order to power these, in order to power all of these reactions. The problem, though, is that I don't think they paid very much attention to almost anything that Nick Lane had to say. They say, for example, that in his book he doesn't discuss homochirality. That's not true. They should read it again. He does, though briefly, discuss homochirality. But another thing that they don't seem to know is that when Nick Lane talks about the potential of acetylphosphate functioning as a much simpler primordial energy currency, he points out that bacteria today use acetylphosphate. And so they act as if this is just a fairy tale with no evidence and it's all just coming from his imagination. Not only do they pass over all the work that him and his group have been doing showing that this is a possibility, but they also ignore the fact that he's pointed out that it works in extant bacteria. And so I wanted to fact check Lane about this. Is it true that bacteria use acetylphosphate to power metabolism? And so I went and I looked, and I found Kuhn et al. 2014, Structural, Kinetic, and Proteomic Characterization of Acetylphosphate-Dependent Bacterial Protein Acetylation. And in the abstract, they write, we detected acetylation of thousands of lysine residues on hundreds of Escherichia coli proteins that participate in diverse and often essential cellular processes, including translation, transcription, and central metabolism. Many of these acetylations were regulated in an acetylphosphate-dependent manner. Mass spectrometry showed that this ACP-dependent acetylation is both non-enzymatic and specific. Crystallographic evidence shows ACP can bind to proteins and active sites and cofactor binding sites, but also potentially anywhere molecules with a phosphate moiety could bind. Finally, we provide evidence that ACP-dependent acetylation can impact the function of critical enzymes, including glyceraldehyde 3-phosphate dehydrogenase, triose phosphate isomerase, and RNA polymerase. And so I had this, I'm going to give you this last quote to finish up here, and I finally have reached the end of it, far longer than I was hoping to go. Thank you again for humoring me. But this is just, it's a closing quote, and it kind of ties into like the whole idea about non-equilibrium thermodynamics, the idea of these energy gradients being fundamental to the emergence of life, driving it into being into a non-random way, and the notion of this kind of power being central to what living organisms really are. And so it comes from the philosopher Friedrich Nietzsche that I mentioned to you before we started the program, and it kind of encapsulates how I look at the universe as a whole and ties this into the origin of life. And so this is my closing quote. And he writes, "And do you know what the world is to me? Shall I show it to you in my mirror? This world, a monster of energy, without beginning, without end, a firm, iron magnitude of force that does not expend itself, but only changes its face. A sea of forces flowing and rushing together, eternally changing, eternally flooding back, with tremendous years of recurrence, with an ebb and a flood of its forms, out of the simplest forms striving toward the most complex, out of the stillest, most rigid, most frozen forms striving toward the hottest, most turbulent, most self-contradictory, and then again returning home to the simple out of this abundance, out of the play of contradictions back to the joy of concord, still affirming itself in this uniformity of its courses and its years, blessing itself as that which must return eternally, as a becoming that knows no satiety, no disgust, no weariness. This, my Dionysian world of eternal self-creation, of eternal self-destruction, this mysterious world of the twofold voluptuousness, my beyond good and evil, without goal, unless the joy of the circle is itself a goal, without will, unless a ring feels goodwill toward itself. Do you want a name for this world, a solution for all of its riddles, a light for you too, ye best concealed, strongest, most intrepid men of the blackest midnight? This world is the will to power, and nothing besides. And even ye yourselves are this will to power, and nothing besides." Friedrich Nietzsche, 1888. Thank you for listening. Appreciate you.
Wow, great job, Drayce. You know,
Thank
you're a really good representative. I feel like you gave a really wonderful presentation. So, thank you. And I feel like, okay, but I have to say this, okay? As you're giving this presentation, it almost feels like you're helping my side. The reason why is because you're actually getting in the weeds of all the things that have to come together
Hell yeah.
for life. Okay.
Yes.
So, I mean, I'm kind of sitting back like, wow, at the end of this, people are supposed to say this all happened and not just randomly, but deterministically, like, because we actually, I feel like we kind of need to go forward with the discussion. I need to understand how you envision what the first cell was like. And Demeter asked this: what is life? And so we kind of need to define what is life. What would you say would be the minimum structure that we can call life?
So usually what I use is like a default model was put forward by Tibor Ganti. It's called the chemoton model. And these are usually called, in the field, they're called the pillars of life. So you have metabolism, compartmentalization, and information replication. And I think that there are certain other things that need to be added to that that have just as, that have a strong claim as well, such as excretion, waste removal. And so you're talking about metabolism, building up and synthesizing organic molecules, breaking them down. You're talking about compartmentalization within a membrane. And you're also talking about informational replication and the excretion of waste. And I think that those, and so we're not simply talking about what life is made out of, we're also talking about what life does, that really defines it for what it is. And so I think that those kinds of pillars are good proxies for understanding what life is.
And do you feel like the steps that you just described are sufficient to produce a cell that, or, if whatever you want to call it, protocell, whatever, that could reproduce, that has information, that is, what are the other things that you said?
reproduce, and it's has metabolic processes going on. It's
Right.
compartmentalized within a membrane.
Right.
And one thing that I didn't talk about, though it was part of my presentation, I skipped over it, was waste removal. Very quickly, how that works is that the waste particulates, they're very small, and so they pass through the membrane very easily. This has to do with like the tomb problem that's mentioned both in Stairway of Life and in general creationist critiques. Like, how is it you need nutrients to get in and you need waste to get out? Well, the actual things that get encapsulated, they're larger. They cannot pass through very easily. The waste particulates, though, are much smaller, and so they pass out. Now you could ask the question, okay, well, they pass out real easy, but doesn't that mean that they can pass right back in? And yes, if they stuck around, but because they're small, they're not big, they are acted on by this process that I talked about before, thermophoresis. So what ends up happening to them is the waste is naturally filtered out. It flows through that alkaline fluid and percolates out into the ocean where these heavier organics are trapped inside the vents in the colder, more stabilizing regions. And that's how this process of this heat gradient drives these organics to the outside and the waste on up and out.
Okay. So how do you envision all of these processes working together in a way that is, I mean, I heard you describe each step, but it's hard to envision how all of this could come together. And, all the information is there, all the, ability to replicate is there, all of the metabolic processes are there, cell membrane is there that has the capability of letting the right things in and sending the right things out. I mean, how do you envision this ever coming together at one time?
Yeah, because it's all taking place in this one environment that is catalyzing this diverse range of chemistry. And the reason why it's able to do that is not just because of this pH gradient between the two fluids, but also because of the wide range of mineral catalysts that are there. So for example, one of the complaints that I didn't talk about, but I think is really important to talk about abiogenesis research, is that they use purified industrial chemicals or minerals. But that's not found in an environment. But as a matter of fact, you don't want there to be just pure iron in this environment, because you're not just trying to catalyze only the things that iron can catalyze. You want to catalyze a range of products, a diversity of chemistry. So you need not just iron, but you also need nickel, and you need molybdenum and tungsten and copper, and like all these different diversity of things, which is actually typically the case in rocks. You'll find all sorts of these different kinds of minerals. And at the vents in particular, you get all of the ones that I just mentioned, as well as zinc. And another important one, even in trace amounts, the work that's been done on this by Harpreet Kaur was like mind-blowing to me, that even in trace amounts of palladium, you synthesize like robust aspects of our metabolism, in terms of like the acetyl-CoA pathway, the citric acid cycle, and reverse Krebs cycle. All of this can be driven, catalyzed, even by trace amounts of this metal. And the clay that I talked about, Fougerite, in certain papers, they refer to it as the poor man's palladium. So I don't know too much about how it's similar, but apparently it is in certain respects.
Okay, well, Drayce, isn't the bigger complaint, though, about the purified chemicals? The bigger complaint is that all of the amino acids and everything used in these origin of life research things, these are all taken from life. Like, we take it from life and resynthesize it and then use it in origin of life experiments, when it doesn't occur. These things in this purified form do not occur outside of life. Like, we took it from life and then did things to it and then put it back in for the experiments. Do you see what I'm saying?
I see what you're saying. So basically how there will be certain scientists, right, who they'll synthesize a certain product, but instead of taking that product and moving it over to the next step of the reaction, they'll purchase it instead and move on to the next step. Is that what you're talking about, maybe?
Yeah.
Because like in terms of like that is that's one aspect where like they're talking about the actual organic molecules that are being synthesized. What I'm talking about here are like the minerals that are like kicking these processes into working, and the thing that I was trying to emphasize there is that you know, the complaint about purity, there's a bunch of things that are in my opinion very wrong with it. The first is that in an actual scenario of abiogenesis, you better hope that it's not pure, because if it is, you're only going to synthesize a very limited range of chemistry. But you need to synthesize this diversity, like you were talking about before. You need all this to happen in this one environment. The only way that you're going to get that to happen is if you have a multiplicity of catalysts that are specialized in different respects to produce this vast range, and that's why I think the hydrothermal vent hypothesis is very good, because it can offer you at the vents all of these different kinds of metals that are able to, as shown in all these different experiments, able to synthesize this vast range of chemistry. Does that answer your question a little bit?
Well, I mean, it was more of just a pointing out that I don't think the biggest problem with the purity aspect is the minerals, but you know, I understand your point about the minerals. So I guess it's just still all of these processes that you're talking about, it's hard to envision how that could end up in, you know, let's say a protein formation without the information. You know, there's the big chicken and egg problem. I mean, it feels like there's a lot of chicken and egg problems, and I've heard you propose some, you know, possible solutions to those, but I don't know. It's still, I don't feel like you've given a complete answer on, you know, where these things could have come from. You know, the complex machinery that's in even the most simple cells.
Heard. So like things that are really complex, like that, for example, the ATP synthase. That's something that is under this model coming way later. It's way too complex from my perspective to be to appear right there at the beginning. But what I did offer was an alternative that's much simpler but does all the same things, which is acetylphosphate, which could also be used as an energy currency. And what they've shown in terms of using acetylphosphate, which is much simpler and which would be available at the vents, is that you can, so far experimentally, they've shown that you can synthesize all the way up to adenosine monophosphate using that. And like I said, like, you know, Herrera took that AMP and under nanofine aqueous conditions synthesized RNA, and gave that presentation to the Europlanetary Society. I always mess up what society it was that he gave his presentation at. But yeah, so like what I'm focusing here on with respect to this model is the experimental evidence that you could use something much simpler than ATP to power these reactions. So like not only is there acetylphosphate in this environment that can power reactions, but there's also pyrophosphate. And the way that pyrophosphate functions is that it stops reverse reactions. So once you have this, once you have a reaction take place that drives it to a greater level of complexity, the pyrophosphate that's there locks it in. It prevents it from reversing back into a simpler state. That's something that that's shown in this paper by Wimmer, published in 2021, Energy at Origins, where a lot of people were at first hoping that pyrophosphate would actually be the simpler energy currency, but she focuses on this analysis of metabolic pathways broadly across life, and what she shows is that in literally 0 reactions is pyrophosphate ever actually used as a currency to drive reactions. What it is used for, however, is to lock reactions into place so that way they don't reverse back to where they were before the start of the reaction. So those two things, acetyl phosphate to power it, and pyrophosphate locks it in. And those two things would be present at the vents with the experimental work to back all that up.
Okay, and then how are you envisioning the membrane enclosing, and like the in the simplest life we have right now, just simple one-celled organism, there's thousands of machines that are working to let certain things come in through the membrane and not let other things come in, and sending out and working as gatekeepers, and all of that has to happen. So you've described some processes like that could create lipids, but how are you envisioning the first membrane, and like how many gatekeepers does it have?
So when I went through my presentation, I talked a bit about how I'm actually a little bit undecided about that, because there's a couple of different options that are really plausible to me. And the first one that I'm going to talk about is one that I really did not like at first, which is the notion of an amyloid membrane. These are basically, instead of having a lipid compartment, it's actually a proteinogenic material that encloses it in, and that it's actually like a peptide that forms a membrane. And the reason why I didn't like this at first is that when you read about amyloids, these are like abnormally shaped proteins that are usually really bad for you. Like there's a lot of research that's done that certain diseases can be caused by productions of like amyloid proteins in your body and stuff. But then, but there are certain researchers that were really pushing this, and I found out a bunch of things that I didn't know before, such that they also have beneficial functions, and that there are even bacteria that today use amyloid proteins for their membranes. And I didn't know that. This was talked about in Duvall 2019, Fougerite, the not so simple progenitor of the first cells. And so that's like, because I can't rule that out, because they've made that sound more plausible to me than I was previously interested in, I'm open to that as a possibility. But in terms of the lipids, once you get a proper, something that's talked about in Boudin and Sostak 2009, is that these amphiphilic vesicles, or basically lipids, they will automatically form a membrane, but in order for you to have that spontaneous process take place, you need to reach a certain critical concentration of them together. And once you get enough of this concentration, they will form this membrane. They call it a critical aggregate concentration, or CAC. And so one of the things they did is they used the thermal gradient that I talked about, where you have this heat source that creates this force and flow away from the heat source and concentrates these molecules together in these colder regions of the vents. And they were able to drive the lipids like over 1,000-fold concentration toward a particular area, in their experimental setup. And when they were driven there and concentrated, they automatically formed into membranes. And not just did they form into membranes, but they also used DNA in their experiment and showed that they encapsulated the DNA. When they reached that critical CAC, they spontaneously formed the membrane and encapsulated the DNA.
Okay, so in either one of those ways that you suggested for forming a membrane, how many gatekeepers do you imagine would need to be there for letting things in and sending things out, considering that our simplest form of life has thousands?
So at first there is likely to be either none, because gaseous nutrients get in past the membrane. Like with amphiphilic vesicles, they are selectively permeable in themselves innately already. So with respect to like gaseous nutrients, they automatically can pass through. and with respect to the waste products, like I was talking about before, because they're so much smaller, they also automatically pass through. And because the flow takes these smaller waste particulates and shuttles them out of the vent, they don't get to stick around to shuttle back into that cell. They get dispensed of. And so the environment is naturally getting rid of that waste that you would need some kind of like, in terms of these intramembrane bounded proteins in order to shuttle things in and push things out. Because of the selective permeability of amphiphilic vesicles and the nature of the nutrients and the waste particulates being what they are, or like, for example, the waste particulates being much smaller, like I was talking about, they naturally pass through. Like Irene Chen is an abiogenesis researcher who talks about this. Her main focus is on a particular kind of membrane that I've read about, but I don't know enough about to really like get into very in depth called coacervates. And like a ton of research has been done on these things, but I've only read a few articles on them because my main interest for a long time was lipid membranes, because that's what most of life uses today. And so I'm interested in finding out, like, in order to gauge the plausibility of a theory for the emergence of life, that theory needs to be as similar to the way that life works today as possible. It can be much simpler, but it needs to still have like the same basic constituents at least. So, for example, it's still at this first setup, you still have a very simple lipid-based membrane with amphiphilic vesicles, but you don't need most of these intramembrane bounded proteins in order to shuttle things in or push things out, because of the nature of the environment and the nature of the selective permeability of the membrane. A lot of that gets handled both environmentally and innately by the nature of the amphiphilic vesicle itself.
Okay, and in that scenario, because we really haven't talked about where the information in DNA could have come from. So how would you envision DNA coming about?
Heard. So there is an article on like a one-pot synthesis of DNA nucleosides that I read recently that was arguing that DNA and also like you get deoxyribose and stuff like that from some of these chemical reactions. So you get the components of DNA from a lot of the chemistry going on at hydrothermal vents, but in terms of like when DNA formed and when you start to get transcription and stuff like that, that's still something that that I'm working on. That's like when I emailed you a while ago, I was talking about like the two things that I wasn't going to touch on very much tonight was like the origin of transcription and the origin of the differences in the membranes between bacteria and archaea. I have like, there's a working hypothesis that I read recently by Victor Soho. And so basically he talks about like, these intramembrane bounded proteins that you're talking about, once proteins start partitioning into the membrane, and I think that like at the origin of life, I said maybe zero, but more realistically, you're probably going to need some few for the purposes of energy harnessing, and so there have been some experiments that I've read where they're able to get peptides to partition, to the membrane, but where was I going with that? I'm sorry, I'm losing my thought. I apologize. Where was I going with that? So you have
formation of DNA.
Okay, so yeah, so there are differences between, so the DNA thing, I don't want to touch too much other than to say that there's experimental evidence, showing that the components of DNA will form under these conditions. But as to the actual emergence of transcription, I don't know. I can bring you as far as good evidence for translation, with protein and RNA. But as far as DNA goes, that's something I haven't read enough about to comment on in any educated way.
Yeah, well, I mean, the issue is not just can the nucleotides form, can they form in homochiral form, and and then can they the transcription happen. Where does the information come from? Because you can have nucleotides that string together and don't mean anything, right, and
Yeah,
don't do anything productive. So
heard.
like this is a gigantic like, so maybe it's something you haven't looked into yet, but this is a
Oh,
huge issue.
so well, actually, that on that particular point, like the whole thing about DNA and transcription is one thing, but the origin of information is another because RNA is an informational molecule, and so with respect to the emergence of information, there's a lot of interesting work, and I wanted to touch on that, but I kind of skipped over it, which I guess I really shouldn't have done because it is a really big issue. Um, but one of the things is there are, oh, hold on, let me see if I can, I always forget the damn name. It's called GARD. Let me see what it stands for. I always forget. Graded Autocatalysis Replication Domain. I think that's what it's called. Oh, hell, I'm not going to find it in 21 pages of notes. But I think it's called Graded Autocatalytic Replication Domain. And what this refers to is this idea that once you have like nucleotides, so you have like a nucleotide sequence contained in a lipid membrane, but those membranes will, through, for example, oscillation with that thermal gradient, so you have this heat source, and as they get close to the heat source, the lipid membrane will break down, but then it actually forms up again. And when it forms up again, it forms longer than it did previously. This is something that's shown by Claudia Bonfio in a relatively recent paper. But the interesting thing about its reformation is that when it reforms with a different sequence of nucleotides. And so the interesting thing about this is that through these gradual processes of like lipid division, you generate this process where all these different nucleotide sequences are being tried out inside these lipid membranes as they divide and reform and stuff like that. So when these different sequences are all being constantly tried out in different ways, that's a way to explore sequence space for the emergence of actual functional information, because you're trying out all of these different sequences as the lipids divide. And so, it searches out for those functional informational sequences.
Okay. And so how frequently do you envision this process was able to happen? Like
And
this to get to that point where these it can seek out or whatever different combinations. I mean, how many tries do you think it got?
a lot. And so like, and I guess the things that I want to stress here in terms of why I find this plausible is that you have these vent systems, like there's not just one or two of them, like they're literally all over the seafloor. And they're lower temperature, they have this chemistry, a wide range of chemistry to produce these things. And so you have multiple different areas that are able to try out these trial and error processes, and the actual trials with the lipids dividing and stuff like that. That's a limited process in the respect that like it'll happen a few times to a particular lipid before it grows large enough that it becomes stable and then partitions into the cold area. So like for each lipid, you'll have a few trials of trying out different sequence space. And then, but again, like you have a huge concentration of these lipids within these vents and many, many different vents across the seafloor. And so you have tons of different simultaneous trials going on of these different sequences being tried out.
Okay, well, I guess I lost my train of thought because I looked over at the side chat, but I'm hearing you say there's lots of tries for each, the lipids and the numerous areas that it could have tried this out. But I guess I'm still incredulous when it comes to this idea that you've got to get all of these systems in place in order for a cell to replicate, right? Like, and I'm just not seeing how in the processes that you've described, this is going to come together in a timely manner for this cell to be able to replicate with having the information for, protein production, whatever that looked like at the time, because maybe you could say these things had simpler, forms or whatever. But I mean, in our simplest cells, there are incredible machines working together, and information for those machines is there in the cell, and it has to be, right? And so I'm trying to think of a simpler form of life, but one that's still able to self-replicate and move on. And so can you describe what that looks like? Like, what does this cell have?
So one of the things that I talked about that facilitates this kind of replication is that
Wait. Hold on.
Okay.
Drayce, I'm not gonna, I don't understand all of the fancy talk. Okay. I'm not gonna know
Yep.
I'm not gonna know. I need to know from you, what does this cell look like in simple terms? Like, does it
Okay.
have proteins? Does it have, what does it have? Does it have DNA? Does it have like in simple terms?
And okay, so what your simple cell looks like based on what I told you, it has a membrane, and that's either going to be an amyloid protein or a lipid membrane. And right now, I would prefer a lipid membrane, even though these other researchers have really, argued persuasively to me for an alternative. But lipid membrane makes, it's more congruent, at least. Well, I can't even argue that anymore, because like when you talk about these, I just have to say this, like when you talk about these proteins embedded in the membrane, they actually make up such a substantial portion of the membrane that these other authors are arguing that we're actually seeing this backward, that we shouldn't be looking at the lipid as being the primary component of the membrane. We should be looking at the proteins as the primary component of the membrane. And that's why they're arguing for originally a protein membrane. But I want to put that aside and just focus on a more classical perspective that where we're using a lipid. And so you have a lipid membrane, and inside that lipid membrane, you have peptides and you have RNA informational polymers. And the interaction between those peptides and that RNA facilitates replication. And and in terms of like, if you have one of these guys who doesn't do anything, they will, over the course of the oscillation in this environment, have sequences of trials where they can potentially pick up a functional sequence. And if they don't, they'll partition and eventually be useless and go out of existence. But over the course of these trials, there will, odds are, there's going to be one out of the many, many countless synonymous trials that take place that develops a robust capacity to replicate with good fidelity. Does that answer your question a little bit more clearly?
Yes, it does. Thank you. Yep. Okay. Are there other topics that you want to talk about related to this? Because we also have audience questions. So
okay. I got out most of my information. I'm sure I'm missing some things, but at this point we're approaching 2 hours. I think I've given enough thought for one night. So
okay.
If you have some audience questions.
Well, actually, I have one more question when you were talking, because you were kind of saying you were saying, I think what you were saying was you are going with a metabolism first rather than RNA first.
Yes.
Is that
yes,
right?
In the sense, but only in the sense that it is a metabolic process that kickstarts the whole thing. But in terms of replicating molecules, like they are there, like I said, RNA, or at least some kind of informational polymer is there pretty much from the onset of life. Like One of the things that I think has really held back abiogenesis research, I think I was talking to you about this a little bit before I went on air, is the fact that so many of them for so long were operating in a vacuum in their own, this came first world. But there's this paper that was published not too long ago, Origin of Life, Bridging Decades Old Divisions, and also John Sutherland has spoken about this as well, where like they came to realize that none of this is occurring in a vacuum, and this diverse range of chemistry is all going on at the same time. And so we really shouldn't speak of like a lipid world or a metabolism first or a replication first, because all of this is happening simultaneously. But ultimately, yes, it's a metabolism first in the sense that it is a metabolic process that's initiated at the vents that kickstarts the synthesis of all of these organics and leads to the complexification of all these things.
Okay. I think one thing that we didn't talk about that was in the Stairway to Life is the problem of having means for repairing biopolymers. Okay,
Yes.
so, we have repair systems that repair everything, DNA, and, repair anything that gets messed up in the cell. Where is where does that come in this model that you're proposing?
So, so there are a bunch of things, like, for example, in Stairway to Life, they point out about how many of these DNA repair enzymes and so forth, how many of them are concentrated on like repair for oxygen damage, or and so like it gave like this huge number that's responsible for that. But here we're in an anaerobic environment. There is no free oxygen at all in this environment, and so there's absolutely no need for these repair enzymes, because it's not getting hurt in that manner. Likewise, because we're either in the ocean or deep in an impact crater lake on a terrestrial continental crust, it's heavily insulated from another kind of potential damage that they didn't explicitly mention in the book, but is also responsible for a lot of damage, which is UV damage from the sun. But because you're in depth in the water, this is already heavily insulating you from damage from the sun. And one of the things that I won't blab too much about tonight is that I think that there's good evidence. I didn't bring up any phylogenetic evidence tonight because I know that most creationists really, they just talk about phylogenetics as just lines on paper, as if it means nothing. But there's a substantial amount of like phylogenetic evidence in favor of this theory that people who already are in favor of things like evolution and who take phylogenetics seriously should really look into. But for me, when I started to look into the phylogenetics of early life, what I found is that there's this this DNA repair enzyme called photolyase that's responsible for UV repair, and it's present in LUCA, which means that it came very early on. And what that suggests to me is that even though this is taking the emergence of life would be taking a place in a place that's heavily insulated from UV damage, it has to be happening somewhere close, because very soon after the origin of life, you see the development of photolyase. And so I'll close answering your question by saying that something I didn't talk about is the emergence of a self-sustaining metabolism. I just focused on like how the environment produces these metabolic pathways. But there's a lot of work that's been done on the synthesis of iron sulfur clusters, which basically manage redox chemistry for biosynthesis. This is what helps you in terms of like respiration and biosynthesis for these organic molecules. This would be the beginning of a self-sustaining metabolism, and they've successfully now synthesized iron sulfur clusters. But when you take a look at something like photolyase, for DNA repair for UV damage, what you find in the most ancient versions is an iron sulfur cluster that's attached to a cysteine residue. and it's actually that iron sulfur cluster with cysteine that's the low um, low quantities of cysteine actually facilitate the formation of these iron sulfur clusters, cysteine being one of those prebiotic amino acids that I talked about before that you get at the vents. And so like you actually see something that biologists talk about a lot is the promiscuity of proteins, that they're able to actually perform many different functions, the same ones, but they're able to do a multiplicity of things. And so you see at the onset, iron sulfur clusters and cysteine doing redox chemistry for respiration and biosynthesis for these organic molecules, but then it gets co-opted for DNA repair for UV damage later on. And so like I would see the gradual emergence of these DNA repair mechanisms, but I don't see them as largely relevant for the origin of life because they're already insulated from a ton of the things that that we now have DNA repair mechanisms for, like UV damage or oxygen damage and so on and so forth. Sorry, that was long-winded.
Cool. Thanks for sharing. Okay, I want to get to some of the audience questions and comments. Peter W. says, is Drayce a biologist? No, but he certainly knows a lot, doesn't he? You should be, Drayce.
It's my GED.
So Chad says, impressive presentation.
Thank you.
Evidence and reason. Sal says, how long are the specific amino acid sequences and the RNA sequences?
Yeah, so there's polymerization of amino acids and RNA. For the RNA, I talked about you get over 200 MERS from thermophoresis. So like you get very long RNA sequences that are capable of doing much more important things. This is something that's talked about Stairway to Life, that and in terms of like Spiegelman's monster and all that stuff. For more information on like how you get these longer polymers, look into what thermophoresis can do, especially that first article that I referenced from MAST, escalation of polymerization at a thermal gradient. This will also work for amino acids and for lipids. The experimental work that I've seen on amino acid polymerization, though, largely doesn't use a thermal gradient. They started in a bit of a mess because what they did was they were synthesizing these amino acids in hydrothermal environments using only the higher temperatures. And so they found that, yeah, we can synthesize all these amino acids, but then they like degrade automatically. And so they were like scratching their heads for a while. And then they were able to, like, for example, take a look at Aubrey 2009, using lower temperatures to synthesize these same amino acids, and they can polymerize. But still weren't making use of the thermal gradient, and I think that that's really important because like the importance of recognizing that in a single environment like this, you have dynamic environmental conditions where you have a hot zone and a cold zone, an oscillation between those two, and it's those kinds of like fluctuations between those two environments that allows for a lot of this complexification to take place. But also with a method like thermophoresis, and this is not the only one, like the one that I focused on tonight only deals with like pretty much low salt environments, but there's a bunch of other ones that these researchers have focused on for much more salty oceans that have to do with like precipitation and electrical interactions and stuff like that. But this particular one drives them, once they sufficiently get long enough, it is these longer polymers that are selectively, preferentially partitioned into the colder regions, and it's in those colder regions that they're much more stable, that they won't degrade because they're no longer by the heat source.
Okay, well, here's a kind of follow-up question to, and this question was posed earlier, so it's not in direct response to what you're saying here, but it is appropriate. Where is the experimental evidence that this leads to life, all these processes that you're talking about?
I mean, like, I think that over the course of the last 2 hours, I just gave it all. Like, yeah,
Right,
like I
but
gave.
in all those experiments, though, we haven't created life, correct?
Yeah, like, no, with respect to this, like there's this whole question of like, okay, well, why can't you create life in a lab? And I don't think that like maybe someday in the future we'll be able to do something like that. But I think that for now, I'm much more interested in focusing on like kind of we've talked before on like the role of the detective, like going back and being able to like take a look at clues at a crime scene and reconstruct what happened. And all of these experiments and all this work in like phylogenetics and etc. Are the clues that you can use to piece together and integrate into a theory. Look at like how events actually operate. What kind of chemistry is involved. The consideration of non-equilibrium systems and like their power generation of driving these things into existence, the capability of harnessing this energy is something that maybe I touched on it a little bit. I can't quite remember, but there's this really important element when it comes to Fougerite. Fougerite, this clay that I mentioned, has all these peculiar capabilities. One of it is that it can get oxidized and reduced, and then oxidized and reduced, such that it's able to harness this energy that's flowing through and pass it along for the onset of these reactions. It is they refer to it as a as a converter and an engine and so on. They use those kinds of terms to facilitate these reactions. But yeah, I just think that like it's I'm not really looking for researchers to recreate life in a lab. Like this is a process that at the very least took 100 years, and I think more plausibly took thousands of years to take place. Something that A Stairway to Life talks about, they insist that it took millions of years, and there are researchers who say that. But if you talk to all of the people today supporting hydrothermal vents, that's not the case. They're dealing with a geological system that lasts for hundreds of thousands of years. And when they talk about these chemical reactions, something that you'll hear Michael Russell say all the time, for a geologist, when you're talking about the emergence of life, convert over to nanoseconds, because it's on the scale of nanoseconds that this chemistry takes place. And so if we're talking about it on this scale, the range that's allowed for at hydrothermal vents is 10 to the 20 nanoseconds, which people like Lane and Russell and so on consider quote unquote plenty of time indeed, since this is the quick rate at which these chemical reactions are taking place. But still, I mean, like that's a very long range of time to recreate in a lab. And besides the fact of like the difficulties of like creating your own hydrothermal vent, like in a laboratory is extremely difficult. Like there's this, they attempted to do it once with this paper Hershey 2014. They had this benchtop reactor, and I was really impressed actually with a lot of the ways in which they tried to build it. But even when they got it to function, they weren't able to get it to function at full capacity, and eventually gave up on it in favor of microfluidic experiments, which are really popular nowadays as a way to simulate vent flow and vent conditions. But yeah, I mean, like it's just a picture that I was really thinking about sending you is a picture of like the vents to show you like their actual size and everything. So there's this picture of like an orca whale, and then there's this picture of a skyscraper, and then there's this picture of the hydrothermal vent, which is just as big as the freaking skyscraper. So if you would like to suggest to me how we can create a lab experiment that involves something like that, I mean, like it sounds to me really difficult to do. The best I think we can do is focus on these clues, analyzing how the vents actually work in modern day vents, their flow and all that stuff, analyzing the chemistry and so on and so forth. That was also very long-winded.
Yeah, you're fine. You're great. Now, does it bother you? But I see all these, you just type in like life from non-life, and like all these articles will come up, like popular media articles that say scientists create life from scratch. And this was even
Yeah.
brought up on one of my streams. So what do you think when you see that?
I just media sucks. It's just like if don't I don't believe anything these days that I read in any newspaper, no matter where it's coming from. Pretty much, like it will depend on like the actual quality of the content of the paper. Like there are some out there that can convey good information, but for the most part, these kind of like sensationalist media articles that you're talking about, yeah, they're garbage. Don't trust what they say. Your best bet would be to dive into the actual literature, which will appear to you like Chinese at first, but there are a lot of ways in which, like, there are many YouTube lecture series by legitimate professors that give you introductory courses into biochemistry, chemistry, physics, thermodynamics. And so you can actually go and watch the actual university courses taught at Yale or Harvard or plenty of other prestigious institutions and learn and pick it up over time. And the papers will become more and more digestible to you.
Yeah, well, some of what you said tonight sounds like Chinese to me, but
All right.
now that I've been exposed to this for a little while, I see what you mean, because at one time it would have all sounded like Chinese, but I've learned a little bit now. But this is kind of an interesting comment. Sal says the asphalt paradox refutes practically all of Drayce's cited experiments. So
Actually,
for those,
sorry, I got to comment on this one. Actually, it refutes none of it because we're operating underwater where asphalt doesn't even form. So like with respect to this aqueous environment, and in terms of the actual experiments, like you're not getting asphalt from these from like this kind of aqueous environment. The asphalt paradox is going to apply much worse to like terrestrial, prebiotic theories. But yeah, the things like the asphalt paradox and the water paradox, I think the hydrothermal vents are uniquely suited to answer that. The water paradox was like its biggest challenge, but in fact, like the biggest challenge only in perception, because people like Michael Russell, who is like the pioneer of this theory from the point of the 90s, like 30 years ago, was already talking about the low water activity that ensues from the confinement that goes on in these vents. And so he actually anticipated this objection long before it even became popular. And all the experimental work that's been done on it by now, like actually demonstrating condensation reaction taking place under these conditions and so forth. But yeah, no, I asked Russell about the asphalt paradox a while ago, and he was, and this was his response too. Is this is something that applies much more for terrestrial abiogenesis theories. This is not something that applies to something that's fully aqueous.
Oh, maybe I'm wrong about what the asphalt paradox is, but I thought it was like basically it didn't have to do with the environment. It just had to do with organic compounds that are left to themselves. Like they just kind of destroy, they just get destroyed. They don't make anything good.
They form this tar. So like, you try to press these synthesis reactions to take place, and a lot of the times what you get is just this tar-like material. But that's not going to, but the response that I received from Russell on this is that doesn't happen underwater. That's something that'll happen, like, Benner talks about the asphalt paradox. He gives this picture of like all this like charcoal, basically charcoal looking stuff that you get. So you can imagine like cooking something on a stove. Looks like you burn the crap out of it, and like that's what you're gonna get. But when you're in like this submersed environment, that's not what you're gonna form, because you're talking about something that's wet. And so that's how I understand that. That is the response that I received from Russell. If Sal Cordova has any further feedback for me on that about why I should be more concerned about the asphalt paradox, because it actually does apply to an aqueous system like this, I'm all ears. But as far as the researchers that I've talked to have responded to me, they said that in this particular environment, that kind of problem will not ensue.
Okay, well, and we can take that up another day. So thank you so much for being here, Drace. You
Thank you for
gave a
having me.
great presentation and I really enjoyed it. I loved hearing your thoughts. And thanks to the audience. You guys have been great too. And do you have any final words?
No, just thank you very much for having me and for having the patience of listening to what unfortunately is still a bit of Chinese, but I'm trying to work on being more clear in my presentation. I was trying to rush it out a little bit, so unfortunately I think I was a little less clear than I wanted to be, but I'm working on getting better.
No, you were as clear could be for someone who knows all the vocabulary. Right? So
Okay.
it had nothing to do with your presentation. Yours, but you did a great job.
Thank
Uh
you.
yeah, I'm just being honest that I didn't understand everything in the presentation, but I thought you came across as very knowledgeable on the things that I don't understand. So anyway, thanks for being here. Good night, everybody.
Thank you. Good night.